Okay, so let's see. So we are going to finish the lecture, the last quarter of the lecture we left out on Tuesday. So this is about whether gene expression noise is related or implies some function in the context of a single cell data. Okay. So we may see noise, and it's white noise, and you can see, okay, so in biology's point of view, so this is something you cannot control. There's a technical noise, and you want to repeat your experiment again and again, try to control. See, I run PCR, qPCR multiple times, take average, I try to control for this variability, the noise. But in here, so in single cell data analysis, we have a specific definition of this noise is the difference in the gene expression level you observed among a group of homogeneous population of cell. So if one cell looks like identical to another, there's no difference in cell type. They cultured in the same petri dish, so in the same condition at exactly the same moment. If a gene is important, you may think, okay, this important gene should be able to express in the same level their dosage effects. So you need to have a similar level of this gene expression in both cells or among the population of cells. But in reality, we always observe some genes do behave like this, like housekeeping genes, as I showed in your example. But some other genes show the opposite pattern. They tend to be very unpredictable, and the one cell may express that gene really, really high, another may shut this gene expression completely down. So it creates this variability. So our job is to, from single cell data, okay, we use different cell lines to extract using informatics approach. Extract those cells which we believe they belong to a group of cells which highly homogeneous in their gene expression pattern overall. So this, in the center of this embedding, the cell distance, very small, they all grouped or clustered in a root location of this embedding. They're highly similar in terms of their overall gene expression profile in terms of thousands of genes you measured. But if you look at individual genes, some will show greater variability compared to another. So that's what we are looking at there within this group of cell. Okay. We look at the different genes. Some genes show uniformly expressed pattern, and that's housekeeping genes or genes that you just indispensable for the cell for living, such as this ribosome protein. If you don't have this subunit, maybe you malfunction in translation, so don't have enough proteins to support activity of cells. Well, this one is not really in essential gene. This is some genes express 3, 4, 600 in UMI count. But as I said, these two have a same level of the mean. So this mu are the same almost. the difference is their standard deviation you measured among the same group of cells, but this immunoglobulin have a really high CV. So that's on the scatter plot. We show the mu and the CV. These two genes have a similar mean but a different CV. This is log scale, so this several times difference. So if we plot all the genes together, some genes have a low expression, average is low here, so but you expect high CV. Some are highly expressed genes, then you expect a low CV. But we are not interested in the absolute value of the CV. We're interested in the relative CV against their expected CV by given mean. Okay, so this is where the difference of the yellow bar here, the distance from the observed to the expectation. So that's called the residual. So you take the difference, absolute difference between these. Those genes, all these genes, which way above this regression curve, show higher than expected variability among them. Okay. So we use this to measure how variable or how noise this gene expressed. If you take, say, 95% of the gene within this region, okay, they are still above this curve, but not extremely high. So we will consider them as a normal, okay. But if you way above, become outlier, we will pick you up. So those are the so-called highly variable genes. So I hope you understand now. We rely on this regression, on the population of genes, to have this prediction model. And based on this model, we found the difference between observed and expectation in order to get this residual CV. Okay? So regression, we know how to do this linear regression. You have multiple linear regression. We can also have a more generalized linear model. So this will be involved in some nonlinear term, involved in the transformation. For example, if I do this log-log, and then you expect a straight line. If you don't do that, you will have some nonlinear, but this, you can do this transformation. There are also linker functions, so this is a more generalized linear model. So you rely on the transformation of your original data to transform values, then expect to have some linear model under the transformed data. So this is a non-linear regression, because here in this plot we do use this non-linear least square, have a curve here. So this is a MATLAB function to do this generalized linear model, okay? So you have all these options, and then you put your x, y data in to, it will have this estimation of your beta values, those coefficients to help you to build this green curve, okay? Okay, so once we have this regression curve, we can see, okay, anything above this curve have a higher than expected variability. So those are the highly variable genes. I have some examples. In contrast, the gene under this curve, this orange regression curve, have a lower than expected CV, so including this ribosome protein and this transmembrane protein encoded gene. Those are the highly variable genes. So this 1, 2, 3, 4, this 2, 1, 2, 3, 4, this 4. These are the two lowly highly variable, lowly variable genes. These two. You can see this magnitude of the Y expression value. So this is from 0 to 10. This is from 0 to 35. So it looks higher bar, but it's just scaled. They all very low, but on average, they have some level of 5 or 10 around here. And they have similar average if you compare them vertically, horizontally, this mean similar. This will have a higher mean, but the most different is their variability. So this cell, for example, have a IGKC have 800, and for this CCL3 you have almost 200 for this cell, but a lot of zeros. So this is highly variable, and those two, these are highly variable genes. Okay, give you some visualization to see this. So this is really high expressed as well. So average is also high, but variability is higher than expectation. So you have FTL gene here. So put those highly variable genes as a group, then we measure, we take a look at what the function as a group they are get involved in. So if you look at the highly variable gene we detect from a lymphoblastoid cell line, and the enriched gene ontology term, including this response, the signaling, transduction, immune response, they are very general terms get involved in the immune system, and also the cytosine immediate signaling pathways. So it looks like everything related to the immune system, and this is a cell which express those genes, and those genes have those functions. Looks like they fit the purpose of this cell. The cell is derived from a B cell. So those are the not marker genes. You cannot rely on this as a marker genes for say B cell because they are highly variable. So if you use this as a marker genes and some cell not express this gene at all, they're not a reliable marker gene. Marker gene, you need to have a consistent, uniform expression exclusively in B cell, for example, not expressed in other cells. So those genes are not marker genes, not a highly expressed gene, but it's highly variable genes. Then look at the lymphoblastoid cell. This is a long airway epithelium cell we used. So our cells will be processed in the same way found. Highly variable, highly homogeneous population, and measure do the regression curve and found those genes. And those genes will have a different name. They are different genes, okay, compared to the lymphoblastoid cell line. But as a group, you can see the function involved in extracellular matrix organization, development stage, the collagen formation. So that's something we found interesting. It also have a cytokine signaling. So those cells, those genes, highly variably expressed in the epithelium cell. In lung airway, they have some immune function as well, but this is more eye-catching, and the collagen formation is definitely will be the function of those cell type. So come back to this fibroblast from skin, okay? We found a different site of this highly variable genes. Those are the gene names, okay? The function you can find more generalized term, development process, anatomical structure development, but also there's a keratinization. So you know it's make the skin become skin, right? Okay. So those terms are unique and should not be messed up with other cell types. So this is very cell type specific. So from these three examples, we can see, okay, highly variable gene will help you to understand what the cell is doing, their job, okay? What the job the cell type is doing. So that's a LCL, so you have a cytokine signaling, and the epithelium cell, you need to have collagen formation. For dermal fibroblast, you have a keratinization. So you don't want to have an epithelium cell have a function of keratinization. That will cause some problem, I feel, okay? You don't want to lymphoblastoid cell line have a collagen formation function. You just cannot mess them up. Okay, so then we look at another group of cells. So as I told you, so we have 3 example cells, but for this, it's slightly different from the others. So the pattern is like this. It's very clean above the curve region. So if you are not stringent enough, none of these genes will be considered as outlier. So there's no highly variable genes in this cell type. So what this cell type is? Can you give a guess, educated guess, or just randomly what do we believe this cell type we are using to do this? So you can try yourself if you found this cell type and download and play with this. It is a stem cell, you're right. So this is a visual comparison. So you can see a lot of those compared to this. This is a stem cell. So our hypothesis here is stem cell has not been differentiated. So there's no function really those cells have been devoted themselves or registered themselves for. So there's no enriched function at all. So you can see from a highly variable gene detection pattern. So if this hypothesis is true, then if we can find a cell type which in the between of the stem cell and the differentiated cell, we should see an intermediate pattern. So you see. So this is stem cell, and this is a differentiated cell type. If we can find a cell type which in between, okay, halfway, you should see this highly variable gene was not there, but it start to show up. So that's what we tried. We tried to use this neural stem cell. So this is a halfway differentiated. You can further differentiate neuron stem cell into astrocytes, oligodendrocytes, neurons, different type of neurons. So this is in the middle. So that's what the neuron stem cell highly variable gene expression pattern looks like. So you can see very clear pattern here, and then start to show some of the genes become highly variable, and then once you become differentiated, fully differentiated, you have more clear patterns in the highly variable gene expression. Okay, so this is a different cell type. So we tried cholangiocytes. Cholangiocytes is special type of endothelium cell, epithelium cell, I don't know, but it's in the in this bile duct, this bile duct membrane. Okay, so the function of this involved in the response to this corticosteroids and this hormone system, which are in the corticoid. So this is really important function, which highly specific to this epithelium cell in the bile duct. If you look at the review papers, you do see this function of those epithelium cell, which what secreting system or hormone system should be getting involved in, and everything makes sense. So this example is tell you it's not only give you the cell type as a large group. You see this epithelium cell, we have a long airway epithelium cell. We also have this cholangiocytes. So highly variable gene give you very, very specific function assignment or classification for the cell type you are under your study. All right. So I think we finished this part. So the take-home message is there are many variability. Okay, there are highly variable genes in there. Compared to housekeeping genes with immunity genes, some of the genes get involved function that require this expression variability. A different cell type have a different group of highly variable genes. And these highly variable genes can mirror or tell you the function of this cell type, such as cytokine and keratinization for fibroblast and dermal fibroblast or cholangiocytes. Okay, so many people don't use this information at all. So if you can use this wisely rather than just compare the average expression of those genes, you may utilize this single cell data more wisely to find a very specific function of the cell. So we are going to go through some of this computational exercise using regression formulas we represent on these slides, and gradually we build up our understanding. Okay, so try highly variable gene detection whenever you have your cell type and you know you need to manipulate to find this highly variable gene in the background of homogeneous population of cell. You cannot say, okay, I have a mixed population of cell, I have a T cell, B cell together, and then you detect highly variable genes. It doesn't make sense because we are talking about the gene expression noise. So you have to apply this to same cell type, same states of cells, as homogeneous as possible of those population. Then from there, you found those highly variable genes using regression method. Then it becomes very informative. Even more informative, you do the same thing for two samples. One is healthy, another one diseased. Then if you found, okay, the highly variable gene changed in the disease states, then you make up your story because some of the function may be lost, some of other function may be gained in the change of the healthy states of the cell population. Okay. So I'm going to go through some of those formulas, code on the slides. Then I will show you the method we developed for highly variable gene comparison in the scenario I just described. Compare healthy with the disease, how you detect those genes. That's variability changed. So it was highly variable in healthy states maybe, and become non-highly variable in disease. Okay? Yeah.
If I understand you perfectly, it means that before you can find highly variable genes, you
Yeah.
need to first, if it's like a tissue attribute sequence, you
Yeah.
have to annotate all the cell types, and you can then run this on the and compare cell type to cell type from different disease states.
Yes, you're right. So you have to be cell type specific. You cannot do this cross cell types. Yeah. Okay. Okay, so let's come back to MATLAB. So I'm in the example. Okay, so I believe we need first develop a function, new function. So we need to have a function to draw a straight line. So you know to draw this straight line, you need for the visualization purpose, because we are going to use data points to find the regression line, and we want to draw on the screen to show the position. So in order to say bx plus c, right? So in order to draw this line, we need two numbers. One is a slope, another is a intercept. So we need a function. If you give me b and c, I will draw this line for you. Okay? So we are going to define this function. In MATLAB, you draw this line. There's two different ways to draw this line. There's a function called line, L-I-N-E. But you need, given the function, 2 points. See, start from here, draw a line, straight line to there. So that's one way to draw the line. We are not going to use this method. We are going to draw a lot of dots which located on this line. We will draw given region from this -5 to 5, this region. Then we will draw dots, follow this line. Okay, if we draw 10 dots, you will see a dot line. If you draw, say, 100 or 200 dots, you will see a straight line. Okay, that's the approach we are going to use. We're going to draw a lot of dots and follow. If you give me B and C, I will, from a region, from this -5 to 5, I will have a -5, -4.9, -4.8. So I will put a lot of dots in there. Then I compute my Y. So I draw those dots on there to reconstruct a line, okay, for visualization purpose. So we are going to develop a function, so called, let's see what the function we should call. Rig line, not very good regress. So let's put I underscore. So I just don't want to make it very general, and so maybe other people develop this function so I have a conflict with them. So I put a I underscore, so that's to build a function with this name. Make it smaller. Where? Okay, so I don't know if you still remember. So I'm in this M file, which it created empty M file, which named this. So I can see a function, and my function will have the same name. I need several input is B, C, and the range from left, right, okay? So that's my function, okay? My L will be the range of the list, and I'll be that. So once I have L and the left and right, this region, range. Let me see lines code. I need to create my X, okay? My X is from left, and each time I will add 0.01 step until get my right. So if you have, say, 4, and then you increase by 0.04 and 0.1 until 6. So if you do this, you will get something like this. Okay, so if you look at my expression here, this is from 4 to 6, and by incremental by step 0.1. If you give this expression to MATLAB, it will give you a series of these incremental steps, from 4 and 4.1, 4.2. So that's what we want. We want to have X, those dots, right? So if we don't fix this starting and end, we replace with L and R. This is not 1, this is L and L, the left boundary and to the right. By small steps, that will create a very dense X, okay? So once we have X is a vector, and we can compute Y. So Y is X times B plus C. Okay, so X may contain 200 points. Depends on how wide you put. If you put very wide, you may have millions of points, whatever X values, but you can compute by multiplied by slope and then plus intercept. That gives you Y. So that will be this line. So now we just need to plot all these dots in the figure. Okay. So we plot x and y using dots, so circle. Okay. That's it. So that's our function, which will draw a line for us. So this function already in the folder, so I can use it. I can change this if you like. If you like a cross, there change the symbol of this. And for the dot, you can have a very thin line there. So this line will be adding into other figures, maybe. So we need a hold on. So you know the hold on. So you hold on the original figure and then add this line onto it. So that's a function we make. Now let's try to use this to plot something out. So I make this, so it's +3. Okay, your every incremental of your, maybe too big, so 1.5. So slope is a positive, then this add 3, and I will do -10 to +10. Okay, so you see this will help you to draw this line here. All right, so we use this small manually created function to revisit how you make your own function, and also see how this line can be drawn. If you do, if you put other symbols, maybe markers, you put an O here, and then you will see different shape, different markers on this line. So if you have circles, it will be many circles. But because it's dense enough, you still see a straight line. So that's what if I change this to O, you'll get. If you zoom in enough, you can see this. If I zoom in here, okay? Zoom in here. You see this. Okay? You can change the color, so into the red. Okay. Okay, with this, we can put this aside. Come back to our examples here. Let's visit my example of this. So now your job is to first draw 3 dots, and you know these dots are from this regression line. Then after you draw 3 dots, then you use that function to do this straight line. So you may want to draw 3 blue dots, and then my straight line across these 3 dots, the regression line will go through them. So that's what your job is. Give it a try. And remember, if you know these 3 dots, you should have a beta 1, beta 0. In our function will be the B and the C. They're fixed. So this is a perfect solution of the regression for these 3 dots which located on the regression line itself. So try this, see if you can make it, and then we will work out if you don't have, if you don't know this line, you don't know the beta 0, beta 1, so we use the equation to compute it, okay, in this perfect solution. Give it a try and let me know. So what the function you need is to do this plot figure and hold on, because you don't want to draw the first points, then the second points, you start with a new figure. So you hold on this figure, then you plot the first dot point by giving x, y. Then you can use a blue circle or something, then you plot the second. So you do this 3 dots through 3 steps. Then you add straight line onto it. So O is a circle and the B is blue, okay? So I do this again, so you don't have to hold on again. So I only hold on once, 0, 0, 2, and 3, 4. So I have 3 dots here, here, and here. Adding my straight line, I will be using the function I just made, and this range will be say -4 to 4. It will be okay. So what is the slope for this example? So the range is -4 to 4, should be okay. The slope, so every 3 increase I have 2, so 2 over 3. And my intercept is 2. Okay. So by looking at my figure, I know the slope so that's a straight line. I know the intercept. I have my solution. Now the problem is so this is a made up, very simple example because I generated those 3 dots by using this regression line on my mind. I know the slope, I know the intercept, made this. But the regression problem is you don't have this slope, you don't have a intercept. How can you compute from the given 3 dots back to there? 3 dots give you x and y, so this is a simple linear regression problem. So from y, from x, from given y, and you try to use x to predict y, and by regress this relationship to find the beta 0 and beta 1. Okay. Beta 0 is intercept c, and beta 1 is a slope. So to solve this, let's see y is what we want to achieve is 4, 4, 2, 0, okay? Okay, our x is, our small x is 3, 0, -3. Yeah, if we centralize y, and then the c will become 0. But in this case, we are not centralized. So we need to put the extra column of 1s next to x. So we will make a big X here by adding by adding 1s against. So how can I add 1? I will do 1, 1, 1, 3, 0, negative 3. So instead of 3 small x, add a 1 to give a chance for the y to move. Otherwise, a y has to be going through 0, 0. So now this problem becomes I need 2 numbers. I need 2 numbers. These 2 numbers will be beta 0 be 0 equals, for example, equals, give just a random number, so 5.6. Beta 1, 3.4. So we need a B which contains beta 0 and beta 1. So we have set up this B, and we don't know whether this is right or not, but just put them in. So if I decide to use a B as a predictor, so B times X, okay? My X is B is 1 by 2 X. Yeah, needed to be 2 by 3. So I think we can 1 by 2 by 3. 2. So what I can do is this times B. Oh no. So I need a transpose in order to make the dot product. Transpose X. Okay, so if I randomly pick a beta 0, beta 1, and then I get this Y prediction, my real Y is this, so it's not a good solution, right? So I need, yeah.
Can you repeat why this is not a good solution?
Because they are very different from my expectation. So this is a computed results, and this is a real I want to fit. So there's a huge difference between them. So I need to correct the beta. I need to change this to two good numbers. I can have a 3 and 5. I try. And compare with my Y. Still not good, so I try another 1.5, 1.3, and I can change this to 2 and I try again. But my Y is supposed to be this. So I can try these combinations of these two intercept and also slope. Basically, I'm trying this different solution until I found one which align onto these 3 dots. So now we know the problems. We have x. We try to find the b combinations of the 2 numbers in order to allow when b dot product with x, you get the perfect y. Okay, so that's our problem. So the solution for this is in order to find the b, so we cannot manually to type and try different. So the easiest solution is to do x / y. Okay. That's the perfect b we will have to solve this problem. Okay, you don't have to try different this dot product again and again. We do x / y. You get the perfect number you have. Let's see whether this makes sense to us. So if I say, okay, this is the b we need, then my b multiplied x, this will give you, still the times b, okay? So this is the y we want to achieve. So let's see again. What our solution for b is this. If you use this 2 times b, and you get a perfect y, okay? Okay, so this is a solution. I think it's time for break, and I show you this is a t-shirt I get from a MATLAB talk. So you see this equation here? Okay, that's exactly it's doing, okay? Just different notations, okay? So this x is our beta, okay? I don't have extra I can not share with you. This is not washed. But I just want to show you. And this is Spanish, I believe. Yeah. Yeah. So remember, the next time you see this, okay, that's what. Okay. Okay, here x and y and b, they are not integers, or they are not single numbers. Otherwise, you can see x divided by b, y, you should get a b, but this will give you a wrong operation. So you have to bring up that particular operators in order in MATLAB. So that operator, if you write that explicitly, and this will be something similar to this, so we can try x negative 1 times b times y. So if you do x, I don't know, can do this and times y. Let me see. I don't think you can do that. Yeah, so it's not operational. Yeah, so we need to a function to have inverse x. Of course. Yeah, so this is require this squared, so we need to borrow this, okay? We need to inverse, because the inverse function will not applicable for this squared, but we can do the p inverse, I believe. Pseudo. Okay, so you do this pseudo inversion, so you get the coefficient the same as what we tried here. Okay. So there are different ways to do this regression to calculate the coefficients. For the more general, it will be do this inverse. This should work because now it become a squared times y. So there's a normal function to help you to get this coefficient. So I just make sure, okay, so you can calculate in different ways, follow those expressions, you'll get the same results at the end. So you see, you can do this because the inversion works on squared matrix. So and this is a more general way to do the fitness. Okay, so if you do, let's try this. This will bring up a function x, y. So we have only one x. So it's very simple regression. This should give you the intercept 2. Where's the slope? Yeah, I don't know how to run this function. So our intercept is 2, that's correct. And for this model, there's something wrong with this because the number is too small. We cannot get a very good estimation for the slope. So for this more general function for the fit for linear model, we need a slightly larger number of samples. Here we only have 3 dots. So the beta estimation is not so significant, so not giving you any estimation. I don't know why exactly like this, but if we try this, so you have a car small and then get X and Y for the car. If you do this copy, so now we are building a more complicated model, have 2 variables to expand Y. So this is a weight and acceleration of the car, and then to predict mileage per gallon, so for Y. So these 2 variables. Two columns give you a prediction of Y in 94 samples. Okay, so this is how you use this function called the fit linear model, which that's inside the statistics toolbox, so in the class. So this function can take larger data set. If you have a huge matrix, you probably cannot do this very effectively. Then you have to rely on this function. They give you more option. But if you look at the results, they are the same. Okay, coefficient. For this relatively small 2-variable regression model. So we also have this regression linear. Remember we had this classification linear. Have your XY from the workspace. So I can have my weight, I need to find a way to get this data in there. So let's see. X and Y, okay. So I will select my X. Response is Y. So this is the way you get the data into here. You use X to predict Y in this 94. And then you start session, and you can select. So that's one of the columns. Okay, this is a 2-variable. So that's a weight against mileage. So you have a heavy car, then your mileage will be worse. And this is acceleration, not so obvious. So you use two variables to explain why. Now you can select, see, I want to linear, and then the linear model will show up for you the training to get the response. You see this for every sample, the original data was that, and then you have Y, which predicted yellow ones. And you can see how good the prediction against reality, and you can calculate the residual of the difference you expected from the observation. So this interface and can help you to build a model for you to save your model for the later use. Okay. There are other more advanced options, tree-based, even use support vector machine to do the support vector regression, etc., etc. So this. I think the next we need to look at the function of lasso. Okay. We know we can have many predictors. See, thousands of predictors to predict Y. Among these several thousands, and which top 10 is most important. We only use 10 to predict, to give me accuracy, which 94% accuracy already. So you don't need to add another 200 or 2,000 variable to improve your. So we need some way to do this regularization. Oh, cannot pronounce this correctly. So let's see where the function is, which lasso is a function in statistics toolbox, which rigid. So this is rigid regression. It's in the same location, same package. So we just need to learn how to run this. So the input will be exactly the same as the regression, because lasso is a regression model, but adding this penalty to number of variables. So if you look at the documentation of lasso, and you will find it looks like exactly the same as regression. Okay, so to regression you have X to predict Y. Here you have the same thing. Let's see doc. Regression is also a function which is the same. Okay. So this is but the XY position is different. The first is Y and another is X, but in lasso is X in the front, Y. They should make this consistent. So this lasso function will return fated least square regression coefficient for linear model, okay, to the prediction of data and to the response of Y. Let's look at example. So for this particular example, the documentation set up a scenario that we generate a data set with 100 samples with 5 variables. That's what this is about. And these 5 variables will not contribute equally to explain why. It gave different weight. The first variable is 0, so basically it's not very important to contribute to the established y value, which second gave you the 2 weight. and the third 0, and the fourth is -3. So there are really only 2 variables in the among 5 of them are really useful. So they set this up and use this weight and the variable to generate the synthetic y with some noise. Okay, so now this setup is like this. You have a sample from 100 data points, and each contains 5 variables. But only variable number 2 and 3 and 4 will help you to explain y. And we can copy this and put that in our example here. I will close the others and then clean my workspace. And then just very clear and just 2 variables, which will be useful in our case. It's x and y, okay? If you do a regression, we get this. So what are these? These are the slopes for each of the contributing variables. We didn't give, should have a slope, should have intercept, which gave extra 1 in the adding to the column. I just didn't do that. But you can see from here, there's some impression that this will not contribute a lot. This not, only these 2 gave you a lot. But we need a better way to do this is to use lasso to give you an x, y, to give you a set of coefficient b. You see, well, this overwhelming, there are a lot of b. I only have 5 variables, and why you gave me so many. So this is some kind of cutoff that you set. I want a very stringent set. I need only 2, then you have only 2. The rest of will be regularized into 0. Okay, they're cut off. If you relax your cutoff, say I want to have 3, then you can include them by relax the cutoff of B. You can move to column. Oh, so for this example, you move into the column. So this is the most stringent. See, 43. So if you only use 1 variable, you have to select the variable number 4. But in most cases, you need 2 variables to explain this. So this cutoff becomes more stringent and more until you get now this variable be included in the model. So this B give you the option. You have a different level of the cutoff. Then you want to very stringent, then you go to the end of big B lambda value to few explain variables. Okay, so this is not a very good example because it's a synthetic small example, but if you have a huge, see, thousands of variables, then this will be very helpful for you to find a cutoff somewhere here to give you 15 variables, major variables to explain your response variable b, y, okay? So for this example, see, found the coefficient vector in the 25. Then you extract the column. From a B, you extract column. So that's your solutions for the problem. If you want to do rigid regression, that's exactly the same, okay? The only difference is how you penalize your coefficient values. So you know this is based on the L2 norm and in the 3, at least 3. I don't know, yeah, so the number of, I believe this is some kind of a number. I don't know how to use this. So what is a K in there? Oh, it's an extra parameter here. Yeah, so you need an extra parameter to fine-tune the some kind of column vector. Oh, yes, y. Oh, so that's a y in front of x. Yeah, so it's opposite. Lasso is x in the front, y in the second, but this regression put the y in the front. Anyway, so that's how we use regularization functions. Okay. So at the end, I'm going to use example data to show you how to do this highly variable gene analysis. First, so to bring up this, we try example data. Okay, there are two highly variable gene analysis. One is a single sample. In this, you will have a group of cells and then found the highly variable gene in that cell group. Another will be two samples, healthy against a disease, and see what's the major change between the highly variable genes and found those that change the most. So let's go to the single sample highly variable gene analysis that will be in here. Found. Yeah, it's in the second item in the analysis. So this function will apply to a group of cells under consideration. But right now, our sample have a multiple cell types, so it doesn't make sense for us to do this. We need to have a single cell type to do this. So that's a warning message. If you still want to do, and you can still keep going, but it doesn't make sense to us based on the principle of this. So we need to we need to focus on only one cell type. So what cell type we have? Click this and label. We have alpha cell. Let's just use this endocrine, okay, endocrine cells. Since we have enough, and also there's a function involved. So how can we extract this cell type? the function you can use is to click this, select by cell type, and we only select endocrine cell lines here. Okay. So now we can close the original because we only focus on this cell type. So this is cell type specific. If I label the cell type, I should have only one cell type. And also, this cell type seems there are subcluster in there. This is not homogeneous, so I'm going to brush and delete this small cluster. Delete selected. So this is relatively homogeneous. I need also have a filter for the cell and the genes, okay? Because you remove a lot of cells, so this cut off. You need to clean the data again. Okay, so this is data is clean, and oh, there another population, so I need to treat them separately. Okay. But for our purpose, I'm not going to further break it down because I end up with not enough cells to show you the process. But in reality, you need to be very, very stringent to clean until you have a homogeneous population. So in this case, for example, I will do the re-embedding, okay? Not use this, but to do this T-SNE 3D again and see whether there are still two populations. I will do this highly variable gene detection separately for even same cell type, different subclusters. Okay, but for the demonstration purpose here, I'm going to use all the cells because this number is too small already. I may end up with no cells to play with. So here, I just decide to run this directly. Use all the cells included. I'm using a spleen fit approach. Okay, so you should be able to get a 3D curve. Okay, so this is a 3D method. So what I showed you in the slides, there are 2D, you have a CV against the mean, but the 3D give you some dimension which you can help you to get better results. So this includes the mean, CV, and the dropout rate. So you can highlight some genes in those regions to find those highly variable genes, which any genes away from this this curve are supposed to be highly variable genes, so the far away the most extreme. Okay, so those may be very important. Remember, our whole population separated into two classes, and this not a very good example to show you the highly variable gene for uniform homogeneous population, because if you look at this example, the one class expresses this gene, another not express this gene. So that's create some kind of artificial, highly variable. That's only because there are two populations. One express CD74, another not expressed. But that's just give you some idea why this position, this CD74 stand out as outlier. But others, maybe you have some other example genes. I don't know if you can do this on your own machine. Can you get this? Okay. So this just help you to visualize those highly variable genes. And those genes really highly expressed there at this end. Okay, so with this we can, let's see what's the first button. You have a button to highlight the top, that's the top 10. So those are supposed to be top 10 highly variable genes. This is to genes from list. So there are some functions for those. So let's see. Genes from this. Yeah, so if you want to see one of the genes, see whether my MRPL4 or this the important, I can see, okay, highlight these two. These two genes seems not very outstanding, so this is a very general. But if you on your mind there's one gene should be very special, you want to know the location of that particular gene, you can select, highlight this where this gene is. We can let's see, enrichment analysis. We can run put the top genes into enricher analysis, and then to see what's the ontology of this top genes I get involved in. So we can see their molecular function, that's including cell components. Remember, we had endothelium cells. Yeah, those are the top function, top molecular function. In the samples, we had this highly variable genes we get from. Okay, any questions? No? Okay. So that's a tool can help you to visualize, to help you to do this. Let's come back to this, see whether we have two samples. If I look at the batch ID, I do have this difference is between different development ages. So I'm going to compare these two to do analysis, which DV, okay, differential variable, highly variable gene detection is for single single populations from single sample. But now we have 2 samples, so we can compare. This is, well, this is human data, right? Yeah, to make this highly informative, I'm going to remove a lot of those less interesting genes, because at the end, I want to, even I see the results from the housekeeping genes maybe not mean anything. So I remove them and make this search space smaller, and then compare the grouping to batches. And that's the results, the difference between so this you see the difference now. You have 2 curves. One is blue and one is orange. And for the same gene, you have 2 profiles. Yeah, so this line, you see this gene, it was, in one sample you have location here, another sample location here. There's some error in there, I check. But some genes, you have a huge jump, okay, in terms of their variability across samples. So this gave you the visualization of their difference. And this is a table for you to summarize. There's a top difference, these genes between 2 samples. Okay. I think it's all for this week. All right. I will see you next week. See you next Thursday. Tuesday, Salim will help give lecture. Okay.